Have you ever gazed at the stars and wondered if time flows differently out there in the vast expanse of space? The idea that humans might age slower in space has captivated our imaginations for decades. This intriguing notion stems from two distinct scientific concepts: the biological effects of spaceflight on the human body and the mind-bending principles of time dilation in physics. In our quest to unravel the mysteries of aging beyond Earth, we must delve into these concepts, exploring the fascinating interplay between biology and physics. Join me on this extraordinary journey as we navigate the intricate workings of the human body in space and venture into the realm of Einstein's revolutionary theories. Together, we will embark on a quest to uncover the truth about aging in space, separating science fiction from scientific reality. Along the way, we'll encounter groundbreaking discoveries, perplexing questions, and the sheer awe-inspiring nature of the cosmos. So, fasten your seatbelts and prepare for liftoff as we explore the fascinating question: do we age differently in space? Space is an unforgiving environment that presents unique challenges to human physiology. The conditions in space are vastly different from those on Earth, and our bodies are not naturally equipped to handle them. Unlike Earth, where gravity anchors us firmly to the ground, space travelers experience a phenomenon known as microgravity, a state of near weightlessness. This lack of gravity affects every aspect of their daily lives and bodily functions. While floating effortlessly might seem appealing at first, prolonged exposure to microgravity can have detrimental effects on the human body. The absence of gravity means that our muscles and bones are not being used as they would be on Earth. One of the most significant concerns is the loss of bone and muscle mass. In space, astronauts must engage in regular exercise to mitigate these effects. But even with rigorous routines, some loss is inevitable. Without the constant pull of gravity, our bones and muscles don't need to work as hard, leading to a gradual weakening over time. This weakening can affect an astronaut's ability to perform tasks and maintain their health. This process is similar to what happens as we age on Earth, but it occurs much more rapidly in space. The accelerated rate of bone and muscle loss in microgravity is a major concern for long-term space missions. Furthermore, space is awash in harmful radiation from the sun and other cosmic sources. This radiation is much more intense than what we experience on Earth. Earth's atmosphere and magnetic field provide us with a protective shield, but astronauts venturing beyond the shield are exposed to much higher levels of radiation. This exposure can have serious health implications. This radiation can damage DNA, increasing the risk of cancer and other health problems. The long-term effects of this exposure are still being studied, but the potential risks are significant. The combined effects of microgravity and radiation pose significant challenges for long-duration space missions, highlighting the need to develop countermeasures to protect astronauts' health and well-being. Scientists and engineers are continually working on new technologies and strategies to mitigate these risks and ensure the safety of those who venture into space. As we delve deeper into the intricacies of aging in space, we encounter two fascinating biological concepts: telomeres and epigenetics. These concepts are not just scientific jargon. They are the very essence of how our cells age and how they might behave differently in the unique environment of space. These microscopic structures and processes play crucial roles in cellular aging, and their behavior in space has sparked both intrigue and debate among scientists. Understanding these processes could unlock new ways to combat aging and improve health, both in space and on Earth. Telomeres are protective caps found at the ends of our chromosomes, akin to the plastic tips on shoelaces that prevent them from fraying. They serve as a buffer zone, protecting the vital genetic information during cell division. With each cell division, our telomeres shorten, eventually reaching a critical length that triggers cellular senescence, a state of irreversible cell cycle arrest. This process is a natural part of aging, but its acceleration or deceleration can have profound implications for health and longevity. This progressive shortening of telomeres is considered a hallmark of aging. It is a key indicator that our cells are reaching the end of their lifespan, and it plays a significant role in age-related diseases. Intriguingly, studies have shown that astronauts' telomeres can lengthen during space missions, suggesting a potential deceleration in cellular aging. This phenomenon has puzzled scientists and opened new avenues of research into how space travel affects the human body. This unexpected finding has led some to believe that space travel might hold the key to slowing down the aging process. The idea that the harsh environment of space could somehow rejuvenate our cells is both exciting and perplexing. However, this effect appears to be temporary, with telomeres returning to their pre-flight lengths upon return to Earth. This suggests that the space environment may induce temporary changes that are not sustained once back in the Earth's gravity. Epigenetics, on the other hand, refers to changes in gene expression that occur without alterations to the underlying DNA sequence. These changes can be influenced by various factors, including environmental conditions and lifestyle choices, like a series of switches that turn genes on or off. Epigenetic modifications influence how our genes are expressed, affecting various biological processes, including aging. These modifications can be reversible, adding another layer of complexity to how we understand aging. Research suggests that space travel may also influence aging on an epigenetic level. The unique conditions of space, such as microgravity and increased radiation, could lead to changes in gene expression that impact aging. The unique environmental stressors encountered in space, such as microgravity and radiation, could trigger epigenetic changes that affect gene expression, potentially impacting aging processes. These stressors create a unique biological environment that we are only beginning to understand. While the precise mechanisms by which telomeres and epigenetics are affected by spaceflight remain elusive, their study holds immense promise for understanding the complexities of aging, both on Earth and beyond. The insights gained from this research could lead to breakthroughs in how we manage aging and improve health in the future. The relentless march of time takes its toll on every organ system in our bodies, and the cardiovascular system is no exception. Our hearts, the vital organs that keep us alive, are constantly working, beating thousands of times each day to pump blood throughout our bodies. As we age, our hearts and blood vessels undergo a series of changes that can increase the risk of cardiovascular disease, a condition that affects millions of people around the globe. Cardiovascular disease remains a leading cause of death worldwide, claiming countless lives each year and placing a significant burden on healthcare systems. In the weightless environment of space, the cardiovascular system faces unique challenges that can mimic some aspects of aging. The absence of gravity affects how blood flows and how the heart functions. Without the constant pull of gravity, our hearts don't have to work as hard to pump blood throughout the body. This can lead to a variety of physiological changes. This reduced workload can lead to a decrease in heart muscle mass and function, a condition known as cardiac atrophy, which is similar to what happens with age-related cardiac atrophy. Moreover, spaceflight can cause changes in blood vessel structure and function. The arteries, which are responsible for carrying oxygen-rich blood away from the heart, can become stiffer and less elastic. This stiffness increases the risk of hypertension and other cardiovascular problems, making it crucial to understand these changes for the health of astronauts. Adding to these concerns is the detrimental effect of space radiation on the cardiovascular system. Space radiation is a significant risk factor that can exacerbate cardiovascular issues. Radiation exposure can damage the lining of blood vessels, leading to inflammation and plaque buildup, which are precursors to serious conditions like heart attack and stroke. These findings underscore the importance of monitoring astronauts' cardiovascular health during and after space missions. Continuous health monitoring can help detect early signs of cardiovascular issues. Developing countermeasures to mitigate the negative effects of spaceflight on the cardiovascular system is crucial for ensuring the safety and well-being of future space travelers. Research and innovation in this field are essential. Scientists and medical professionals are working tirelessly to develop strategies and technologies that can protect the cardiovascular health of astronauts. From specialized exercise regimens to advanced medical monitoring equipment, these efforts aim to counteract the adverse effects of space travel on the heart and blood vessels. As humanity looks towards longer missions and potential colonization of other planets, understanding and protecting cardiovascular health in space becomes even more critical. The knowledge gained from studying the cardiovascular system in space can also provide valuable insights into how we can better manage and treat cardiovascular diseases on Earth. These advancements not only benefit astronauts but also have the potential to improve cardiovascular health for people worldwide. The race against time continues, but with dedicated research and innovative solutions, we can hope to safeguard our hearts, whether on Earth or in the vast expanse of space.